Hello everyone, I am going to share a research article published in Nature, which is about gut microbial metabolism contributes to insulin resistance. I am Shazia, working in diabetes and cardiometabolic disorders lab in Dr. Fazirabi Aman's group. So, this is the brief introduction of this research article. What is the insulin resistance and the role of gut microbial insulin resistance? Insulin resistance is the primary pathophysiology underlying metabolic syndrome and type 2 diabetes. The human colonic microbiota is a large and complex microbial community. The gut microbiota has been implicated in host energy metabolism and metabolic diseases. The gut microbiota break down various nutrients, produce metabolites that play a fundamental roles in host metabolism. This is the background of this research. Metagenomic study have described the characteristics of gut microbiota and their roles in metabolizing major nutrients in insulin resistance and insulin sensitivity. Carbohydrate metabolism cells has been proposed to contribute up to 10% of the host overall energy extraction, thereby playing a role in the pathogenesis of obesity and prediabetes. So the hypothesis behind this research was gut microbiota might play a role in insulin insulin resistance. So this is the overview of this research. Total 306 individuals without a prior diagnosis of diabetes, diabetic medications, or intestinal diseases were included. Insulin resistance and metabolic syndrome were the main clinical phenotypes. The host microbe relationship, host factors such as clinical plasma, metabolome, peripheral blood mononuclear cells, transcriptome, and cytokines data were collected. Similarly, microbial factors such as 16S virus sequencing, shotgun metagenome, and fecal metabolome was also gathered in this study. This workflow shows the multi-omics analysis. A phenotype-associated metabolomic signatures by winning metabolites into coabundance group were analyzed. Microbial signature were determined using the and metagenomic data sets, and their association with metabolites were analyzed. To gain insight into the host-microbe relationship, the association among fecal metabolites, microbes, and host plasma metabolites, cytokines, and peripheral blood mononuclear cells genes were analyzed. The mediation effects of plasma cytokines on the relationship between fecal metabolites and metabolic markers were also assessed. For the validation of candidate bacteria, metabolites, bacterial culture, and animal experiments were performed. So it comes to results. This graph showing that fecal carbohydrate metabolites are distinctly altered in the insulin resistance. To examine how omics data of fecal samples can predict insulin resistance, area under the curve of characteristic curves on the basis of random forest classifiers were compared. Predictor variables for the models were selected using the minimum redundancy, maximum relevance algorithms from the fecal 16S ribosomal RNA, metabolome, metagenome, and their merged data sets. The selected features of the fecal metabolomic data generally outperformed and metagenomics in predicting insulin resistance, suggesting that fecal metabolomics could be used to study insulin resistance pathogenesis. Among the hydrophilic metabolites, most of the coabundance group showing the significant association with insulin resistance were those of carbohydrate metabolites, mainly monosaccharides such as hydrophilic coabundance group 5, 12, and 15. Among these major monosaccharides such as fructose, lactose, mannose, and xylose significantly correlated with insulin resistance. Additionally, short chain fatty acid, which are also known as carbohydrate ferment, were also increased in insulin resistance, such as the hydrophilic coabundance group 8 that is mentioned. Among the short-chain fatty acids, propionate was particularly increased in insulin resistance, consistent with its role in gluconeogenesis. Hydrophilic coabundance group remained unannotated as it included metabolites from different pathways. In addition to hydrophilic metabolites, fecal lipid coabundance group were also associated with insulin resistance. Among them, a lipid coabundance group largely consisting of diglycosyl, diglycosyrol, which is abbreviated as DGDG, is the major lipid, CAG11, which is mentioned here. That is the interest of this study. It is reported derived from the bacteria. These lipids contain glucose, lactose in their structures. Most of the DGDGs in their clusters showed positive correlation with some of the precursors, diacylglycerols and monosaccharides, and that is the glucose and lactose. As diacylglycerols are deeply involved in the insulin resistance pathogenesis, the biological function of metabolites, these metabolites are the particular interest of this study. Notably, the DGDGs with different acyl chains in lipids, such as covalent group 41, showed no association with insulin resistance. That implying that differences in acyl may have a physiological importance as reported previously. So move towards the next results. This graph showing the insulin resistance associated fecal metabolites that are associated with altered gut microbiota and microbial genetic functions. The study participants were classified into 4 clusters: cluster A, cluster B, cluster C, and cluster D, that are on the basis of their taxonomic profiles. Individuals in cluster C distinctly possess group 2 with bacteroideals, whereas those in cluster D higher abundance of group 1 and group 3 bacteria. Notably, the proportion of insulin resistance was significantly lower in cluster C, and the proportion of insulin resistance among individuals with abundant group 1 and 3 bacteria was consistently higher than those with abundant group 2 bacteria, as identified on the basis of Futkan metagenomics data. So the detailed analysis of cage orthologs. The other metabolic parameters associated with insulin resistance and metabolic syndrome, such as homeostatic model assessment, insulin resistance, BMI, HDL cholesterol, and adiponectin were also different between cluster C, with the lowest proportion of the insulin resistance, and the other 3 clusters. Insulin resistance associated fecal carbohydrate predominantly correlated with the genera in group 1 and group 4. The most prominent being Doria in Lechnospiraceae. So by contrast, the majority of these carbohydrates negatively correlated with the insulin sensitivity associated genera in group 2 bacteria, such as bacteroids, Aedes types, and Phyllominifractors. So detailed analysis of GG orthologs revealed that fecal carbohydrates and participant clusters mainly correlated with phosphotransferase system, that is abbreviated as PTS, relating to disaccharides and amino sugars. These suggesting that the preference of sugar used by microbiota of PTS may affect the metabolites level. Glycosidases were also associated with the fecal monosaccharides, and extracellular glucosidases such as beta-fructoferrosidase and amylosucrase and oligoglucosidases, which are predicted as a to degrade the sucrose and dextrine into glucose and fructose. So these showed the highest positive correlation, especially with the fecal glucose. Importantly, the abundance of these glycosidase genes was significant between participant cluster C and other three clusters, suggesting that the taxonomic profiles largely explain the variation of glucosidases. So consistently disaccharides breakdown genes were predominantly conserved in the genomes of Brutia and Doria, abundant in cluster D, whereas they were almost lacking in bacteroideals, abundant in cluster C. So move towards the next slide. This slide showing the fecal carbohydrate metabolites that are associated with the cytokine levels in insulin resistance. From the cross-omics correlation-based network analyses, it could be seen that fecal carbohydrates were strongly tied with both bacteria and host insulin related signatures, especially cytokines, suggesting that these metabolites are the hubs of host microorganisms network in insulin resistance. The most pronounced association was between the fecal carbohydrates and cytokines. Notably, interleukin 10, which is mentioned here, interleukin 10, plasma cytokines showed the most prominent association with carbohydrates, and the other cytokines such as adiponectin, leptin, serpin E1 also showed an association to lesser extent, and the modestly with PBMC derived transcripts, supporting recent studies showing its paradoxical effects to facilitate insulin resistance. So the causal analyses were performed to check the cytokines mediated effects of fecal carbohydrates on host metabolism. It has been found that interleukin 10, serpin E1, adiponectin, and leptin mediated most in silico causal relationship between fecal carbohydrates and post insulin such as HOMA-IR, such as the HOMA-IR. Notably, there were unique correspondences between the metabolites and cytokines. For example, interleukin 10 mediated the effect of fructose, mannose, xylose, and rhamnose, but no other, not so this slide showing that insulin sensitivity associated bacteria ameliorate insulin resistance in experimental models. The above finding from human multi-omics analysis revealed an association between carbohydrates and insulin resistance pathology. To address the causal relationship between gut microbiota, fecal carbohydrate, and metabolic diseases, 22 human fecal insulin sensitivity and insulin resistant associated bacteria were cultured. And the top 10 metabolites contributing to the group, including several amino acids and the fermentation products such as succinate and fumarate. And the majority of these metabolites were preferentially produced by bacteroideals. They detected the 13 out of 15 carbohydrate associated with insulin resistance in the bacterial culture. Most of these were plotted negatively along the PC1, suggesting that these metabolites were negatively associated with bacteroideals. Glucose, mannose, and glucosamine were preferentially consumed by bacteroideals family. Insulin tolerance tests also revealed that this trend insulin resistance most prominently by Alice types in distinctus administration. So this is the conclusion and all about of this study. This metabolome-based approach identified related to insulin resistance, association between fecal carbohydrate and low-grade inflammation of insulin resistance, and efficiently select candidate strains for functional validation in experimental settings. So this study also highlights the advantage of a comprehensive omics strategy in exploring the involvement of microbial metabolism and their product of insulin resistance. In vivo studies confirm that this Aristides and distinctus administration improves lipid accumulation and thereby insulin resistance while simultaneously reducing intestinal monosaccharides levels. Everyone for the attention.